This is an overview of an article entitled A Proteomic Analysis of the Myocardium in Hypertrophic Obstructive Cardiomyopathy, published in the December 2018 issue of Circulation, Genomic and Precision Medicine. Hypertrophic cardiomyopathy, or HCM, is a common inherited disease caused mainly by dominantly inherited mutations in genes encoding cardiac sarcomere proteins. The disease phenotype is characteristically diverse, ranging from no clinically detectable abnormality to extreme hypertrophy and death from ventricular arrhythmia, heart failure, and stroke. The biological effects of individual genetic variants only partly explain this complex clinical spectrum, and much still needs to be learned about the downstream pathways that lead to disease development and progression. Global proteome studies have provided mechanistic insights into many human cardiovascular diseases, including heart failure, aortic aneurysm, atherosclerosis, atrial fibrillation, and inflammatory dilated cardiomyopathy, but there have been few proteomic studies in HCM. In the paper under discussion, the authors performed comparative whole-tissue proteomics using myocardial tissue removed at the time of septal myectomy. The aim was to identify dysregulated proteins that provide insight into disease pathogenesis. Label-free proteomic analysis was used initially to compare protein profiles in myocardial samples from 11 patients with HCM undergoing surgical myectomy, with control samples from 6 healthy unused donor hearts. Differentially expressed proteins of interest were validated in myocardial samples from 65 unrelated individuals by the development and use of targeted multiple reaction monitoring, or MRM-based triple quadrupole mass spectrometry. A total of 1,586 proteins were identified across all heart samples. Principal component analysis demonstrated that the control samples were clustered with separation from the HCM samples, indicating the HCM tissue had a distinctly different proteome. Significant differences between HCM and controls were observed for 151 proteins, with 56 proteins being upregulated and 95 downregulated in the HCM samples. Gene ontology and pathway analysis indicated that many of the differentially expressed proteins were associated with metabolism, muscle contraction, calcium regulation, and oxidative stress, as shown here. The authors used MRM mass spectrometry to assess 33 of the previously identified proteins in the validation cohort, which comprised mostly HCM samples with smaller numbers of control samples and samples from aortic stenosis patients. They found a total of 8 proteins to be significantly altered between the HCM and control groups, including the proteins indicated in the graphs shown here. The discovery of significantly elevated levels of lumacan was a novel finding and considered to be relevant to disease mechanisms. Therefore, 2 distinct lumacan peptides were chosen for inclusion in the validation study, with a strong correlation observed between their levels, as shown in panel A. Lumacan concentrations were higher in HCM samples, correlated with left atrial area on echocardiogram, were higher in 16 patients with a pathogenic sarcomere mutation than 24 patients with no pathogenic mutation identified, and were higher in those with late gadolinium enhancement, or LGE, in the septum on MRI, compared to those without LGE. This last observation suggests a connection with myocardial fibrosis. Carbonic anhydrase 3, or CA3, concentrations were higher in HCM samples, but showed no difference between the patients with a pathogenic sarcomere mutation and the patients with no pathogenic mutation identified, as well as no difference with respect to LGE on MRI. As shown in the lower panels, the CA3 protein was present in the myocardium, with increased expression of CA3 in HCM samples being confirmed by immunohistochemistry, where it was found to be localized to the cytoplasm of the cardiomyocytes, with considerable heterogeneity in expression both between samples and within the same sample. In conclusion, out of a total of 1,586 identified proteins, 151 proteins were differentially expressed in HCM samples compared to control samples, with many of the proteins associated with metabolism, muscle contraction, calcium regulation, and oxidative stress. Thus, the myocardial proteome of HCM provides supporting evidence for dysregulation of metabolic and structural proteins. Finally, the finding that lumacan is raised in HCM hearts provides possible insight into the myocardial fibrosis that characterizes this disease.